So I would like to talk about single cell data and using it in order to make inference about tumor phylogenies. And before explaining a little bit our approach, I will make a few comments on why we might be interested in doing that and what exactly I mean by this. So the starting point in more general terms, as you are all aware, is that we're not using the drugs that we have available in a very efficient way. This is a rather old study, but nevertheless, it shows you the percentage of patients for which a certain drug, stratified by classes, has not worked, has not done its job. And without going into any details here, for cancer drugs, that is this amazing number of 75%. So I don't want to argue about the number, but it's clear that what we're doing currently here is suboptimal. And the hope is, of course, or one hope at least, is that we can stratify patients in a more detailed and in a better way and use improved molecular diagnostics in order to predict whether a certain patient will respond or not to a treatment, and in a similar way, for example, whether the patient will develop severe side effects or not. So there's many approaches to doing these molecular diagnostics, and I'm interested a lot in sequencing, DNA and also RNA sequencing. And we're doing this not only, but a major attempt is in the context of oncology. So there we're working on the computational backbone for the molecular tumor board. So the molecular tumor board, at the end of the day, is just a group of experts that decides on the treatment together. And if you start, well, you start, of course, with biopsy material, and then you're doing diagnostic tests, for example, based on DNA sequencing, then it's actually a long way to get these sequencing results into the tumor board. And some of the steps here in between and some of the reasons why this way is long and difficult to go, I want to talk about today. So major steps here include variant calling, so actually detecting changes in the DNA relative to a normal genome. That already turns out to be very difficult in general, as I will show. And then, of course, you need to interpret these genetic changes. And while for a few mutations and a few genes we might know what the effect is, there's a vast number of genetic alterations in cancer genomes. And for most of these changes, it is very difficult to predict what their effect on the phenotype and eventually on treatment outcome would be. So the way we are looking at cancer is very much from an evolutionary point of view. So cancer is an evolutionary process, more precisely a somatic evolutionary process. So the cells in the tissue replicate, they divide, and of course they need to copy their DNA, and there's an occasion there for mutations to accumulate. And some of these mutations may give rise to a fitness advantage, to a growth advantage of a cell, and then the clone that grows here may acquire additional mutations. So overall, we're interested in this process of tumor progression, and in particular, the genetic basis of this process. This is just another view of the same process where the size of the clone is on this y-axis. And this is supposed to be the first cell that is altered, genetically altered. And this clone starts to grow, and you see new mutations coming up. Some may go extinct, and others continue to grow and eventually dominate the population. Now, the data that we will have available is a snapshot at one point in time. So you're taking a biopsy, and that means you're sampling this diverse set of tumor cells at a certain point in time. And the tumor phylogeny problem now really is to take a sample from the cells at this one time point and to reconstruct the evolutionary history of the tumor. So that means we're trying to identify the different clones that are present in the population and their relationships. So we're trying to reconstruct what the common ancestors of these clones are and what the branching structure of this phylogenetic tree is. So let me continue by making a few remarks why this tumor phylogeny problem might be interesting. It's certainly interesting from an evolutionary biology point of view, but for this audience that might not be the best motivation. So the first thing is that we can distinguish different models of tumor evolution. There's a lot of discussion in the field of how this process works.